Evguenia, thanks for coming on the show this morning. Very nice to have you. Now, you're
Thanks for having
working
me.
on cancer, right?
Yes, right
Okay.
now I do cancer research.
What aspect?
We study some proteins that normally protect us from cancer.
We have proteins that protect us?
Yes,
Well, that's a good thing.
because there's lots of damage that we all have in our everyday life, and we need proteins that protect us all the time. They're called tumor suppressor proteins. Otherwise, people would have cancer early on. But we have these special proteins.
All over the body, in all our cells?
Yes, in all our cells. One of them is called P53. That's
It's
the
called
protein.
what?
P53.
P53.
That's a simple name. Yeah, but unfortunately, this protein can get mutated itself, and then those cells are not protected from cancer anymore. And our recent research found that also this protein actively promotes cancer, unfortunately. So we're trying to fight it.
Oh, wait a minute. So the defender turns into an attacker
Yes,
somehow?
exactly.
Why? How does that
That's
happen?
horrible. It just gets mutated itself. And in fact, it is mutated in estimated half of all cancer cases in human patients. So it's a very important protein that when mutated gets really bad.
When you say mutated, what do you mean?
It just had, so a protein has a sequence of amino acids, and when one of them is substituted, then the protein maybe has a different shape and can't do its job anymore.
All right, so let me understand this. So there's a protein that we have in all our cells whose normal job is to
Protect us.
But
Basically,
fend
what
off
it does, it senses stress in the cell. If something goes wrong, if other genes are broken apart, then what it does, P53, it causes the cell to repair itself. And the second job that it does, if a cell cannot repair itself, for example, there is too much damage now, then this protein tells the cell to self-destruct. So that cell is eliminated early on without becoming a cancerous cell.
So it normally does a good job, but sometimes if it gets
Exactly.
damaged, it starts to do a bad
Exactly.
job. It makes things
Exactly.
even worse, do you imply?
Absolutely, it makes things really bad.
How?
When it's mutated, that cell is not protected and becomes a cancer cell that divides and gives rise to an actual tumor.
But you said that if the
And
guarding protein goes wrong, it's worse than if it doesn't have it at all?
Exactly. If this protein is mutated, one might think that it's just not working. But what recent research found that now it actively promotes cancer, and we are trying to establish mechanisms why it now becomes a really bad guy. It wasn't known 10 years ago why, and so very recent research. And what we're trying to do, what people tried to do before, was to revert the mutated P53 into normal and hoped that now it would stop the cancer. And people were not successful just technically in doing that. So what we wanted to do, since we discovered that it's a bad guy doing really bad active job, we are trying just to eliminate this mutated P53, even without restoring the good guy. Just by eliminating the bad guy, we wanted to see, will that stop cancer? And we do that in mice, and it works.
It works?
It
So
works.
By just taking it away
By
somehow?
taking it away, even without providing the good version.
Ah,
Yes.
so we don't really need it in most cases then?
Well, I guess there are other proteins that, without it, other tumor suppressor proteins that substitutes for a job.
Just not
And
so
that's true.
good.
It's not the only one. There are many, luckily.
So can we use this? Is it
Yes,
available now? To
absolutely.
treat
Right
people?
now, we tried it in mice.
In mice.
And in mice, what we did is we removed the mutant P53 gene because we designed a mouse in a special way that we can excise the gene. We cannot do it in humans because our genes are normal. They are not designed in a special way. Now, a second approach we tried is to use an existing drug to remove now the protein in the mice. And that can be directly applied to humans. And so far it's been working in the mice as well. So it's very promising. We need to initiate clinical trials, but
we've heard lots of drugs work in mice and don't work in people.
that's
a big
another problem. That's another problem. That's why all the clinical trials start with very few patients for toxicities, and then if they...
Are you ready to start clinical trials? with
No, because unfortunately our mouse model has a cancer that's very rare in humans. And the effect of the drug might be cancer specific. That's very common actually. So now we are going to test our two approaches in a mouse that has breast cancer, similar to human. If that works, then we will be ready to start clinical trials in human patients.
Well, good luck. I hope you're successful.
Thank you so much.